In wildlife conservation, innovative research and technology push the boundaries of what's possible. Traditional methodologies lay the foundation, while advanced techniques complete the bigger picture and help to fill in the missing details. DNA, an organism's genetic makeup, can tell us a lot about an animal or a population. This information was often difficult to obtain, requiring blood or tissue samples from animals in remote or largely inaccessible habitats. Environmental DNA, or eDNA, is changing that. This non-invasive and expansive approach allows researchers to obtain genetic information from different animals and organisms from tiny particles in the water, soil, and air. Researchers are using eDNA to track endangered species, analyze populations, and tell the whole story of an ecosystem.
DNA stands for deoxyribonucleic acid, and it is a molecule that forms the foundation of life. Every organism on Earth contains DNA.
The combinations of those nucleotides make all of us who we are, as people, as animals, as plants, and the subtle ways that those are different makes us unique.
DNA is what makes me different from you, us different from dogs and giraffes or snails. It's basically the genetic blueprint that makes organisms what they are.
DNA is the genetic code for any living organism, and every species has a separate genetic code. In other words, you can distinguish species by their DNA when we analyze their DNA in the laboratory.
So because DNA exists in all organisms on Earth and wildlife, we can use DNA as a tool to understand, for example, wildlife diseases. We can screen for DNA of organisms that cause diseases like bacteria and fungi and viruses. We can also use DNA to understand kinship and heredity, to really understand how species are related to each other or within a population of relatedness. We can also use DNA to understand from sort of a forensic perspective or a DNA detective perspective of what organisms may exist in an environment. Like, for example, if you're out and you find poop or feces sitting on a forest floor, you can collect that sample and isolate the DNA to figure out what species that sample came from. So DNA is really useful sort of across the conservation field to tell us a lot about the health of the environment, the health of the wildlife, and we use it for all sorts of different projects.
Environmental DNA is DNA that has been shed into the environment by an animal or a plant. So that can be anything from mucus to feces to bodily fluids or skin or hair. Anything that carries genetic material that comes off into the environment can then be collected and analyzed to identify the animals that are there.
That DNA can be collected from water, air, and soil. All right? DNA is everywhere, all around us, everywhere.
I always like to use the example, let's say you go swimming in a body of water. You're shedding cells off your body that is getting into the water. We can go to bodies of water to look for rare and threatened species by just collecting water samples, filtering the water, and then collecting the DNA on filters, and then testing those samples to see if a particular species of interest might be in the environment.
So the New York Seascape program is using eDNA in two different ways. So one way is here on the Bronx River. We're using it to track the migration timing of alewife, or river herring.
We're asking ecological questions in order to help us better understand how we would conserve these species. So for example, we want to know, when are these animals here? What time of year? Are they here all year long? Where along the river are they living? Do they have certain preferred habitats? We can go and collect water samples from those different places, and we'll have a much better understanding about the wildlife that is dependent on this river system.
We are tracking the entire spawning season of alewife. So we go out and we collect water samples, collecting environmental DNA, before the alewife arrive in the system in early spring. And then we take collections all throughout the year.
These animals only come into the river to reproduce and then go back into the ocean to spend the rest of their lives, right? So it's a way for us to track the spatial and temporal distribution of a given species.
The idea of that is we are trying to connect our environmental DNA data with a traditional survey method, which is visual surveys. And for that, we're using an ROV, a remote operated vehicle. We drive it underwater and we look for alewife underwater, and we count the number of alewife that we see, and we hope to be able to correlate that to the amount of environmental eDNA that we collect.
Back almost 9 years ago, when we started first getting into eDNA testing, we were really skeptical. We're thinking, okay, if you collect water samples, really, how well does this work? Can you really detect an organism? And so we went out to our pond on zoo grounds and collected water samples, and we were surprised to find we could identify a lot of the species of turtles within those pond samples. This led to a larger project we embarked upon in 2016, which was to develop a DNA test that could detect this critically endangered turtle species called the Yangtze giant softshell turtle, Raphidius swinoi. And this is the most endangered turtle in the world. There are 2 animals that are known to exist. Because so much effort has to be put into visual observations of this animal, all the resources are really going to the lakes where it's known to exist. And there are many lakes that have not been surveyed very well that this animal could still exist. So we developed an eDNA test specifically for Raphidius swinoi so that we could go and systematically test these other lakes to determine if this animal may exist in some of these areas that hadn't been surveyed.
But we also use eDNA in another way. We use it in what we call eDNA metabarcoding. And that is a tool by which we can actually understand or study the ecological composition, the species diversity in a given location or habitat.
We can collect a water sample, and we can actually see the entire array of life that is present in that water sample.
So in the New York Bight, which in the waters from these tidal rivers like the Bronx River, all the way in through the estuaries, out along the continental shelf, and even into the Hudson Canyon, where we're doing a lot of work, we are able to understand what is the species composition, what is the species richness, or what we often call biodiversity. Each of those areas are going to have a different species composition, different species richness. And by comparing those water samples, we can get an idea of both the spatial, across the Bight, and the temporal seasons, spring, summer, winter, fall, of what species are here and when. And then how do we go about protecting them when they are here from a lot of the human uses that we engage in here in the New York Bight.
The field is changing fast. It's improving with more researchers that are trying to understand the limitations of the technology. So I think there are a lot of ways that this approach can be expanded to different conservation projects and contexts. And so I think this is a tool that's not going to be going away. If anything, it's going to be expanding its use, especially as the technology gets cheaper, and it's much faster to collect eDNA samples than doing some of these traditional surveys, which makes it just such a valuable approach and tool in combination with other ways of monitoring wildlife in an ecosystem.